For thousands of years, cancer was a mystery. Doctors could see the tumors, but they had no way to stop them. In the 1940s, something changed. During World War II, scientists discovered that a chemical used in warfare could destroy rapidly dividing cells. That discovery led to the first modern cancer drugs, chemotherapy. For decades, chemotherapy became the front line in the war against cancer. Tumors could shrink, but the treatment often weakened the body itself. Today, millions of patients still rely on those same strategies, but some researchers believe the real weapon against cancer may have been inside the human body all along, a type of immune cell designed to hunt and destroy abnormal cells. They're called natural killer cells, and their story may reveal one of the most controversial questions in modern medicine. Did we overlook the body's own cure? This is the Scanner Files.
You know, last year, Vice President Biden said that with a new moonshot, America can cure cancer. Last month, he worked with this Congress to give scientists at the National Institutes of Health the strongest resources that they've had in over a decade. So tonight I'm announcing a new national effort to get it done. And because he's gone to the mat for all of us on so many issues over the past 40 years, I'm putting Joe in charge of mission control. For the loved ones we've all lost, for the families that we can still save, let's make America the country that cures cancer once and for all. What do you say, Joe? Amen.
For thousands of years, cancer was one of medicine's greatest mysteries. Doctors could see the tumors, they could feel them growing inside the body, but there was almost nothing they could do to stop them. Surgery was the only option. If the tumor could be removed, the patient might survive. If it could not, the disease usually continued to grow. By the early 1900s, scientists began searching for a chemical treatment, something that could destroy cancer cells without destroying the patient. The breakthrough came during World War II. Researchers studying a chemical weapon called nitrogen mustard noticed something strange. Soldiers exposed to the gas experienced a dramatic drop in white blood cells. Doctors realized the chemical was attacking rapidly dividing cells. Cancer cells divide rapidly. In 1942, doctors at Yale used a form of nitrogen mustard to treat a patient with lymphoma. The tumor shrank. This was the beginning of chemotherapy. Over the next decades, chemotherapy became the foundation of modern cancer treatment. New drugs were developed. Combinations of drugs were tested. Entire medical specialties were built around fighting cancer with powerful chemicals. Some cancers began to show remarkable improvement. Childhood leukemia survival rates rose dramatically. Hodgkin's lymphoma, once considered fatal, became one of the most treatable cancers. But the treatment came with a cost. Chemotherapy attacks rapidly dividing cells. Cancer cells divide rapidly. But so do many healthy cells in the human body. Hair follicles divide rapidly. The lining of the stomach and intestines divides rapidly. Bone marrow cells divide rapidly. When chemotherapy enters the body, it cannot always distinguish between the two. Patients often lose their hair. They suffer severe fatigue. Their immune systems become weakened, sometimes dangerously so. For some cancers, chemotherapy works very well. For others, the results are far less certain. Even today, doctors often describe cancer treatment as a war. A war fought with surgery, radiation, and chemical drugs designed to destroy tumors before they spread. But in recent years, some researchers began asking a different question. What if the most powerful weapon against cancer is not a drug at all? What if the human body already contains cells designed to hunt and destroy cancer? These cells exist inside the immune system. They move through the bloodstream and tissues, constantly searching for abnormal cells. Scientists call them natural killer cells. Natural killer cells are part of the body's first line of defense. They can recognize infected cells. They can recognize damaged cells. And under certain conditions, they can recognize cancer. When a natural killer cell finds a dangerous target, it attaches to the cell and releases toxic proteins. Within minutes, the target cell begins to break apart. For decades, scientists believed these immune cells might play a role in controlling cancer, but activating them in the human body proved far more difficult than expected. Then, in the 1990s, researchers studying immune signaling molecules made a discovery. Inside laboratories at the National Institutes of Health, scientists identified a powerful molecule that could stimulate natural killer cells. The molecule was called interleukin-15. Interleukin-15 acts like a signal. When immune cells detect it, they begin to multiply and activate. Natural killer cells become stronger, T cells become more aggressive. The immune system begins to mobilize. In laboratory experiments, the results were striking. Immune cells exposed to IL-15 expanded rapidly. Their ability to destroy abnormal cells increased. Some researchers began to wonder if this molecule could eventually be used to help the immune system fight cancer. But turning a discovery in a laboratory into a real medical treatment is rarely simple. Years of research followed. Clinical trials were slow. Scientists struggled to find safe ways to deliver the molecule in the human body. For a long time, the discovery remained mostly inside scientific journals. Then, decades later, a surgeon and biotechnology entrepreneur began arguing that the immune system itself might hold the key to fighting cancer. His name was Patrick Soon-Shiong, and his ideas would eventually intersect with one of the largest cancer research initiatives ever announced by the United States government.
Because we have a cell in our body called the natural killer cell that has been born to recognize cells that are without MHC. It's called missing self. And by so doing, that natural killer cell targets all these MHC lost cells and activates a thing called gamma interferon and stimulates back the MHC1. We as physicians, scientists, and the industry almost anticipate progression. We want to change that bar, where it should be cancer-free overall survival. And what we're now seeing in our bladder cancer patients, both in the naive and the BCG unresponsive, that we actually not only have a complete remission, but a durable complete remission. So as I said, time matters, duration matters. In our naive setting, we have now patients we've been followed for up to 9 years with complete remission or disease-free, depending whether it's cyst or papillary, up to over 8.5 years.
By the mid-2000s, cancer had become one of the leading causes of death worldwide. Millions of patients were diagnosed every year. Despite decades of research, many forms of cancer remained extremely difficult to treat. In 2015, the issue became deeply personal inside the White House. Beau Biden, the son of Vice President Joe Biden, was battling glioblastoma, an aggressive form of brain cancer. The disease moves quickly and is notoriously resistant to treatment. Around this time, a surgeon and biotechnology entrepreneur named Patrick Soon-Shiong was invited to Washington. Soon-Shiong had built a reputation as an innovator in cancer research and biotechnology. According to his own account, he was asked to review Beau Biden's medical case. By the time the meeting happened, he later said, it was already too late to change the course of the illness. Beau Biden passed away in May of 2015. His death had a profound impact on his father. In the months that followed, Vice President Biden began advocating for a new national effort to accelerate cancer research. The following year, during the State of the Union address, President Barack Obama announced a major initiative. The program would become known as the Cancer Moonshot. Its goal was ambitious: accelerate cancer research and achieve a decade of progress in just a few years. Billions of dollars would be directed toward research, data sharing, and new therapies. The initiative was placed under the leadership of Vice President Joe Biden. Around the same time, Patrick Soon-Shiong was promoting a different vision for fighting cancer. Instead of focusing primarily on destroying tumors with drugs or radiation, he believed the immune system itself could become the main weapon against the disease. His work focused on a specific type of immune cell: natural killer cells. These cells, he argued, already possess the ability to detect and destroy cancer. The challenge was finding a way to activate them. Soon-Shiong began developing therapies designed to stimulate these immune cells using signals similar to interleukin-15. His approach aimed to expand and strengthen the body's own cancer-fighting cells. At the same time, he launched a large private research initiative involving multiple pharmaceutical companies and cancer centers. The project was also described as a moonshot, but the relationship between this private initiative and the government's cancer moonshot soon became unclear. Sun Xiang has said in interviews that his ideas helped inspire the national program. He also claims that after the initiative began, his role in the effort changed dramatically. According to his account, the program moved in a different direction. Other researchers and institutions took the lead in defining its priorities. The government's cancer moonshot continued to fund numerous research programs across the country. But Sun Xiang's work on natural killer cells remained largely outside the official framework. Whether this was a disagreement over strategy, funding priorities, or scientific direction remains debated. What is clear is that the idea of using the immune system to fight cancer was gaining momentum across the scientific community, and natural killer cells were beginning to attract serious attention.
I've obviously been working in this space for the past 2 decades. I think we've established proof of principle with NK cell therapy, but I think what is going on now is that there's many flavors. My colleague Katie Rizvani, who is talking this afternoon as well, I think was the first in a New England Journal of Medicine paper to put in a CAR-19 into an NK cell product. If you look at the industry involvement in the NK cell field, it's just kind of exploding right now. The last time I counted, there were somewhere between 12 to 20 NK cell companies, with various flavors, whether they be iPS-derived, umbilical cord blood-derived, peripheral blood-derived NK cell lines. And I think now that we could do gene modifications with safety, it's really opened up a whole new field. The T cell field has been probably about 5 years more advanced than the NK cell field. I think, as I talk about NK cells a lot, the challenge, I think, to the NK cell industry is to try to have something commercialized in the next 3 to 5 years. The main selling point about NK cells is they're easier to manufacture as an off-the-shelf product, and they don't have graft versus host disease. In all the literature published so far, they should have a lower propensity to give cytokine release syndrome and neurotoxicity.
To understand the theory behind this treatment, we need to look more closely at the immune system itself. The human immune system is made up of many different types of cells. Some produce antibodies, some coordinate immune responses, others attack infected or abnormal cells directly. Among these defenders is a group known as natural killer cells. Natural killer cells, often called NK cells, act as one of the immune system's first responders. Unlike other immune cells, they do not require prior exposure to a disease to begin attacking. Instead, they patrol the body continuously, searching for cells that appear abnormal or damaged. When an NK cell encounters a suspicious target, it attaches itself to that cell and releases toxic proteins. These proteins puncture the cell membrane and trigger a process known as apoptosis, a form of programmed cell death. Within minutes, the damaged cell begins to break apart. In laboratory studies, NK cells have shown the ability to destroy virus-infected cells, abnormal cells, and in some cases, cancer cells. But the activity of these cells is not constant. Their strength depends on chemical signals inside the body. One of the most important of those signals is a molecule called interleukin-15. Interleukin-15 acts like a messenger. When immune cells detect this signal, they begin to multiply and activate. Natural killer cells expand in number. T cells increase their activity. The immune system shifts into a more aggressive defensive state. Scientists studying this molecule discovered that IL-15 could dramatically expand populations of NK cells in laboratory experiments. The idea soon emerged that these immune cells might be able to attack cancer more effectively if properly activated. But translating this idea into a real treatment proved difficult. Cytokines like IL-15 are powerful signals. In the human body, they can trigger intense immune reactions. Delivering them safely required years of research and experimentation. Eventually, biotechnology researchers began developing modified versions designed to stimulate the immune system without overwhelming it. One of these approaches became a therapy called Anktiva. The treatment is designed to mimic the activating effects of IL-15 and expand populations of natural killer cells and cytotoxic T cells. Instead of attacking tumors directly with chemical drugs, the therapy attempts to strengthen the immune system itself. In 2024, the United States Food and Drug Administration approved Anktiva for a specific type of bladder cancer known as non-muscle invasive bladder cancer. The approval marked one of the first therapies built around this immune activation approach. Supporters of the treatment argue that this strategy represents a major shift in cancer therapy. Rather than suppressing the immune system, they believe treatments like this could help the body fight cancer more effectively. But not everyone agrees that the evidence is strong enough to support broader use.
So the question is, what does Anktiva do? Well, Anktiva, and you can see that from the label, is a first-in-class next-generation immunotherapy that activates NK cells, reactivates, restores CD8 killer T cells, and most importantly, activates the memory T cell. And in so doing, this so-called triangle offense allows us now to kill those recalcitrant, resistant MHC negative cell, which we now made them MHC positive.
Regulators typically require large randomized clinical trials before approving treatments for multiple cancers. These trials can take years to complete and involve thousands of patients. Critics of the regulatory process argue that this timeline slows access to promising therapies.
Extremely healthy. All of this is because of this treatment.
So here's what you haven't been able to fix, which is the demand.
10,000 desperate requests daily. If you look at this one, September 26th, doctor, please help me. It's very hard for us literally to play God. Our drug is approved, believe it or not, just for a tiny piece of a subset of bladder cancer.
That's right. As of today, Anktiva is not available for patients with every other type of cancer: breast, lung, brain, you name it. For that to happen, the FDA says it needs more trials, more data, more proof of safety and effectiveness, and more time. Why is it that the clinicians or the FDA or whatever the standard is not impressed by this?
I think we stuck in the old assumptions. We stuck in the check the box, and we stuck in show me chemotherapy. They don't recognize that this is a ubiquitous treatment of your immune system. You shouldn't ask what cancer is that. That's what we're trying to get the FDA to understand.
Supporters of the process say these safeguards are necessary to protect patients from ineffective or unsafe treatments.
We were fortunate to get a guest who was a former associate commissioner of the FDA, okay? And his name is Peter Pitts, to help us understand the process and the questions that may be asked in response to what Dr. Pat is saying. Hey, Pete, thank you very much for helping us out. Appreciate it. So Dr. Pat's main assertion is this. Look, when they want it to happen, they make it happen for big pharma with other drugs. And when they don't want to make it happen, there's never enough data, there's never enough testing. And Dr. Pat contends that that's the group that he's in, that there's nobody pushing the FDA to make this more available, so it's not happening.
Well, thanks, Chris. Thanks for letting me set the record straight. First of all, once a drug is approved by the FDA for even a small condition, it can be used by doctors for whatever they want. So right now, any doctor treating any type of cancer patient in the United States can prescribe his drug for whatever reason he or she sees fit. So this isn't a question that the FDA is stopping the use of this drug for broader use. This could be a question that insurance companies don't want to pay for it. That certainly could be a
that's
problem.
right.
But I think one of the major
But that's the
issues
same thing, though, Pete.
No,
but hold
it's-
on, Pete. Help me unpack that. Help me unpack
Sure.
that point for people. So, okay, you're my doctor, and you say, yeah, no, I heard about this. I saw the special with Cuomo on about Dr. Pat, but they won't pay for me to have it, and it's very expensive. So practically, you're in the same position. If they don't approve it for more cancers, the insurance company won't pay.
Well, yes and no, because as it stands right now, a lot of insurance companies won't pay for a drug that is indicated for a specific condition. They just deny it. They let people fail on cheaper, older drugs until they die. And that's unacceptable. That's got to change. And that's because the healthcare system doesn't work together. We're focused on getting it approved. That's hard enough. That's a science-based, evidence-based proposition. And then when it comes to paying for the drug, it's an entirely different proposition. It's a new playing field. It's not an even playing field, and patients suffer. Patients and government and advocates and scientists need to focus on getting drugs that exist to patients. And in fact, the FDA has a program called Expanded Access that facilitates and really accelerates getting unapproved experimental drugs to patients. And insurance companies don't want to pay for that either. So it's, one hand is not talking to the other. And unfortunately, the government is not helping, because you have agencies, science agencies, that want to get these drugs to patients before they're approved and after they're approved, fighting the people that don't want to pay for those drugs. And that's the travesty here, that it comes down to not wanting to get people the drugs they can have because they don't want to pay for them.
The debate over how quickly new cancer therapies should reach patients continues today. For patients facing advanced cancer, these decisions are often deeply personal. Many are willing to try experimental treatments in the hope of gaining more time. Others prefer therapies with long records of clinical evidence. As immunotherapy continues to evolve, researchers are exploring many different ways to activate the immune system against cancer. Natural killer cells remain one of the most intriguing possibilities. But whether this approach will transform cancer treatment on a large scale is still an open question. The debate over experimental treatments raises another difficult question. Who should ultimately decide when a patient is allowed to try a new therapy? In the United States, the Food and Drug Administration requires extensive clinical trials before approving drugs for widespread use. These trials are designed to determine safety, effectiveness, and long-term risk. The process often takes many years. Supporters of these safeguards say they protect patients from treatments that may be ineffective or harmful. But critics argue that for people facing terminal illnesses, time may be the one thing they no longer have. Some patients ask a simple question. If someone is diagnosed with an aggressive cancer and standard treatments have failed, should that patient have the right to choose an experimental therapy, fully understanding the risks involved? In recent years, laws known as right to try have attempted to address this issue. These laws allow certain patients with life-threatening conditions to request access to experimental drugs that have passed early safety testing but are not yet fully approved. Even so, access to these treatments remains limited. The issue became even more visible during the COVID-19 pandemic. Faced with a global emergency, governments and regulators accelerated vaccine and treatment development at unprecedented speed. Clinical trials that might normally take years were conducted in months under emergency authorization systems. Supporters say this rapid response helped save millions of lives. But the experience also revealed how quickly medical innovation can move when society considers a threat urgent enough. Cancer has existed for generations. Millions of people are diagnosed every year. Yet, many cancer therapies still require decades of research and regulatory review before reaching patients. For those facing terminal illness, the question becomes deeply personal. If a patient understands the risks and is willing to try an experimental treatment, should that decision belong to the institutions that regulate medicine, or to the patient whose life is on the line? There may not be an easy answer. But for many people living with cancer, it remains one of the most important questions in modern medicine. Cancer research has come a long way since the first chemotherapy experiments of the 1940s. Survival rates for many cancers have improved dramatically, and new therapies continue to emerge from laboratories around the world. Immunotherapy, including treatments that activate natural killer cells, represents one of the newest directions in that fight. Scientists are still studying how these immune responses work and how they can be safely used against different types of cancer. Some researchers believe the immune system may hold the key to future breakthroughs. Others caution that promising laboratory results do not always translate into effective treatments for large numbers of patients. Meanwhile, the regulatory process designed to protect patients continues to move carefully and methodically, often requiring years of testing before new drugs become widely available. For many patients, those years can feel like an eternity. Some will place their hopes in established therapies backed by decades of research. Others will look toward experimental treatments that promise a new approach to fighting the disease. The question of how quickly those treatments should reach patients remains deeply debated among doctors, scientists, regulators, and the patients themselves. Perhaps the most difficult question of all is the one that still remains unanswered. If the human body already contains cells capable of destroying cancer, how close are we to learning how to use them? For now, the answer remains uncertain, and the file remains open.
We were chatting briefly, and you said you had some news to announce, particularly around pancreatic cancer. What can you tell us?
Well, today and tomorrow we'll be announcing at this healthcare conference that we have now, with our natural killer cell, which we've engineered, a complete response in the patient with metastatic pancreatic cancer that had failed all other treatment.
This has been the Scanner Files, scanning the world for anomalies. Until next time.